Hi, this is Ilari from On Humans with one more bonus episode, an unheard clip from my conversations from last year. This one has to do with biology, evolution, and the philosophy around how we think about those things. The target here is nothing more or less than Richard Dawkins' monumentally successful book, The Selfish Gene, and more broadly, the kind of thinking that book popularized. There's really two things in Dawkins' book that I think have struck the imagination and the ire of philosophically inclined readers around the world. One is Dawkins' claim that we are born selfish. Now, if that bothers you, you might be happy to hear that Dawkins actually said that was a rogue claim that you should, quote, mentally delete. I've written about this in more length. You can head to episode 20 to hear me read my essay or get a link to read my essay on the topic. But there's another part in Selfish Gene which is a significantly more open to debate, which is significantly more plausible, let's put it that way, which Dawkins has never retracted. And this is his claim that, and I quote, "We are survival machines, robot vehicles blindly programmed to preserve the selfish molecules known as genes," unquote. What Dawkins really is saying here is that if we take the achievements of modern biology, which is often called the modern synthesis, seriously, what we notice is that evolution is a science that deserves to be there together with physics and chemistry and whatnot. But it is a science of genes. It is not a science of you or me. We organisms don't really matter. We are an epiphenomenon, as the philosophers might say. Or, as he said more provocatively, we are machines designed for a purpose, for a rather ignoble purpose of spreading our genes. Now, there are evolutionary biologists who strongly reject this view on various grounds. If you're interested in it, you can Google more about so-called extended evolutionary synthesis to see what you find there. I talked with one of the pioneers of this so-called extended synthesis, Eva Jablonka. And I had her on for another topic. We talked about the evolution of consciousness. You can listen to that in episode 36. But after we finished that really epic tour, I asked her if she would have a little bit more stamina to stay and discuss her thoughts about evolution beyond the selfish gene. And Jablonka is known for her work on epigenetic inheritance. Epigenetics means beyond genes, after genes. So what does this mean? What do we know about it scientifically? And does it change anything philosophically? It was a privilege to get to ask these questions from Jablonka herself. This was a fun little chat. I hope that you enjoy this. If you do, as always, subscribe to On Humans. Give it a 5-star review if you haven't done so yet. And of course, you can decide to join the wonderful group of people who also support me financially by heading to patreon.com/onhumans. Thank you to all of my Patreon supporters for the support last year. And without further ado, I bring to you all, one more time, Eva Jablonka.
Let me give you an example. Think about a system of transmission of a musical score. How do you transmit a musical score? You copy it. Okay? This is like DNA. Now imagine that you have a new technology, recording and broadcasting. Now what you can do with this technology, you can copy a particular performance. Before you can copy only the score, not the performance itself, right?
Oh, very interesting. When genes copy, they are basically copying the score. Yeah, maybe there's some mutation errors, but the score gets transmitted. The way that a musician has been improvising on it doesn't leave a track on the score for the next generation. And what you're saying is with the epigenetics, it's if there is an improvisation by a musician, that improvisation gets transmitted.
Can be
transmitted because of the recording energy.
That's right.
That's a really nice analogy. Okay.
And now, one of the things that people were thinking about was, how is it the case that the determined cells, the stem cells of your liver and the stem cells of your kidneys are giving rise to liver and kidney? How is it possible? The genes are the same, right? In your body, exactly the same DNA sequence. And nevertheless, this cell divides and gives rise to liver cells. This cell divides with the same DNA and gives rise to kidney cells. And another cell divides and gives rise to your pancreas. And another cell divides and gives rise to your skin cells. They're all the same in terms of DNA. What they're different in is in terms of their gene expression, how the genes are expressed. So they're different in phenotype. So there must be some kind of inheritance system that leads to the reproduction of patterns of gene activity, right? This is epigenetic inheritance within the body. In the 1970s, 1980s, people started experimenting and trying to understand this kind of inheritance of patterns of gene activity in cell lines within the body, not between bodies. And they discovered that there are all kinds of specific and dedicated systems of inheritance that are superimposed on DNA replication. We call it epigenetic inheritance. Now, people thought that this epigenetic inheritance only happens within the body. In the germ cells, in the sex cells, it's all deleted. It's like you have a delete button and you start again.
Can I ask, just so to make sure that I understand right, so sticking to the earlier analogy, would it be like saying that within a certain kind of, say, musical club, yes, the way one performer performs, it will impact the way the next performer performs it. However, between musical clubs, the only thing they ever send each other are the musical notations, and so all the improvisation information is lost.
Well, it could well be that sometimes they meet and the two clubs meet and then they say, wow, look at that. And they transmit it.
Yeah, and this was thought to not take place in inheritance, in biology.
That's right. But let's just think about it like this. People said, you have to build the whole body with different cells. So you cannot just take the memory of one particular cell. It cannot be.
Yes.
It's impossible. It must be all deleted, right? So when we started working on it, we realized that this biology just doesn't work like this because we inherited something additional to the DNA sequence. For example, a certain pattern or something superimposed on the DNA methylation, which is a slow group, or all kinds of other things. I don't want to go into the details because these are biochemical details. There are several such systems. And we realized that this has to be that it will be sometimes not so infrequently transmitted from one generation to the next in a slightly messy way because it will depend on the environment. And the reason that it is important is that it depends on the environment because gene expression is dependent on the environment. It changes as a result of environmental inputs.
Could you give an example of either in humans or other animals where how does this work?
Yes. So let me give you an example, a nasty example. There are chemicals that are called endocrine disruptors. They disrupt the hormonal system. We produce a lot of them. A lot of plastics that we produce are this kind of things, and they cause a lot of problems. They cause all kinds of problems. They reduce fertility. They increase the probability of certain cancers. They lead to obesity, and they lead to all kinds of immune problems. And also some of them also lead to all kinds of cognitive problems. We live in a very nasty, toxic world that is our own creation. Anyway, I will give you an example of an experiment on rodents, on mice. So you give a pregnant female this kind of stuff, this kind of one of this endocrine disruptor, it's called bisphenol A. You expose her during her pregnancy. So you expose the embryos the embryos to this stuff. And as a result of that, they are born with all kinds of problems. Now you take the males with the problems and you make them with females that are completely normal, that has never seen this bisphenol A, were never exposed to it, and then their mothers were not exposed to it. They're okay. And then you look at the next generation. And what you find is they have problems. They're obese, they have all kinds of problems. So now you take again this offspring, and you make them as again normal females that had never seen this. And you look at the next generation, and they have problems. Up to the sixth generation, they will have increased obesity, for example. And up to the fourth generation, they will have other problems. How did it go?
Do I remember right that there is a similar study with mice where instead of giving a nasty chemical, the pups got nasty treatment?
Well, this is another example. So in this experiment, what they did was during the first 14 days after birth, the mother was separated from the pups, from the little ones, for 3 hours a day. And during these 3 hours, and they never knew when, so it was unpredictable. And during these 3 hours, the mother was also stressed in an unpredictable way. She was either forced to swim or she was put into a confined place. So when she came back to the little ones, she was very nervous and very stressed herself. So she didn't give them the best.
This is very not a nice experiment.
Terrible, very, very cool. And what happened was, of course, that the little mice became very neurotic in many ways, right? They became, they showed the equivalent, the mouse equivalent of depression and all kinds of things. And this was translated, it was a whole package of problems that were inherited from one generation to the next, including physiological problems, like sugar metabolism was also fucked up in this mice. And this transmitted for 5 generations.
5 generations, okay.
Yeah, already 5 years. There was a recent paper that is showing already of some of this package. Not all. Not, it's a complicated thing, and this is epigenetic inheritance between generations. Now, why is it important? Because it is the inheritance of acquired character, right? It's a kind of Lamarckian inheritance, right?
Which was not supposed to happen.
At the theoretical level. But why is it important practically? If humans, we are not mice, okay? And it's very difficult to do the translation from mice to humans. We are very long-lived, we're much more plastic. We know that these chemicals are very bad for us too. There is a lot of data about that. And we also know that they affect the next generation. Now, we don't know how many generations they affect and what exactly the mechanism is in the case of humans. We know in the case of mice, we know in the case of rats, and because they're experimental animals that are easy to work with. Yeah, but we are similar enough, but the effects that we know of in humans are similar enough. So this is very dangerous.
Of course, in the nice ways that if we can fix any of these problems, give a good early start to the next generation. Not only are we benefiting them, but perhaps the generations after them too.
We can do it when we give enriched environments. For example, to those poor mice that were neglected, we give them a lot of toys, and we give them a lot of exercise, and we give them a lot of friends, and all this kind of things. Then we change the epigenetic marks. We change the epigenetic.
Really?
Yes, they don't transmit it anymore.
Well, that's wonderful. That's excellent news. I'm glad you added that. But I do want to ask one question on the theoretical level. I mean, I think the practical implications are easy to understand because what we care about is the next generation and generation after that, perhaps. But what about evolution? Because evolutionarily, I think that there are many people who speak for the orthodoxy, who would say that, oh, this is very interesting. Now we have learned that these things can be passed on up to 6 generations, but that's not what evolution cares about. Evolution cares about hundreds of generations. And so still, if we look at how speciation happens, how new species emerge, if we are interested in Darwin's original questions, we still really should just zoom in to the genes, because yes, there are these interesting things happening on top of that, but they are not stable enough, because we're talking about something like 4 to 6 generations, not something that will become a fixed part of the lineage. What do you think of that?
I think it's wrong for many reasons. So first of all, the first reason is that I was talking, for example, about mammals. And plants, for example, it can be transmitted from many more generations. So that's one thing that we have to remember. That's just one thing. The other thing is that we know that even if we have just 4 generations of transmission, first of all, in the same way that usually the conditions of life that lead to the selection of that gene or that or another gene are not just 1 generation or 3 generations or 4 generations even, but many generations, right? Otherwise, there would not be selection. So if the change conditions that induce different gene activity and different epigenetic marks persist, then the epigenetic mark will also persist. And the other thing that is different between epigenetics and genetics is that you can induce, whether a mutation is usually in one individual, you can induce an epigenetic change in many individuals at the same time by the same environmental conditions. But there are several things that one has to understand. First of all, there is an interesting connection between genetics and epigenetics. Because the epigenetic state of a gene, the things that are superimposed on the DNA, change the mutability of the DNA. Makes it more or less mutable. It changes the rate in which DNA changes itself. So, for example, in a recent paper from not so recent already, I think 2021, I don't remember, on a plant, Arabidopsis, it transpired that a lot of in many genes, active genes, the changes in DNA can be best explained by the mutability bias that was created by the epigenetic state of that. I will give you another example. People looked at sea bass. Sea bass is a fish. They grow them in some kind of big cages in the ocean, right? And then we eat them. And they looked at two populations, one population that was kept in these cages for 25 years and one that was kept just for 1 year. And now, as a result of living in this kind of way, there are a lot of changes that are happening at the phenotypic level, at the level of the traits that we see, the macroscopic traits that we see. And this is the result of changes in gene expression. And in the population that lived in this domesticating conditions for one generation, the changes were not in DNA, they were in the epigenetic factors. When they looked, those that were 25 years later, they found that there were mutations in these very genes, in these very regions that were epigenetically modified in the 1 year. This is showing you that there is a relationship between the epigenetic, between a persistent epigenetic state and mutability. This is number 2. Number 3 is that these epigenetic changes can lead to incompatibility. If you have one population living here, even living in one environment, another population living in another environment, genetic changes didn't happen yet, but epigenetic changes are happened in one generation, in one population, and in another population. They come together, and they cannot produce offspring because the epigenetic changes in the germline make them incompatible. So it can affect speciation, and so on and so forth. I wrote a lot about this. So that in which epigenetics can affect evolution.
Now, how revolutionary is this? I guess, so this is my final question, is that this is often discussed as a kind of part of the so-called extended evolutionary synthesis, which is supposed to be an update or even a revolutionary update over the so-called modern synthesis, which is perhaps best, or certainly is best popularized in Richard Dawkins' Selfish Gene. How much change is for the average person who isn't really deeply invested in the exact mechanisms of evolution? The only book they read is Richard Dawkins' Selfish Gene, or not even read it. They've read half of it and heard something about it. How much does this really change the kind of fundamental picture where the organism is kind of almost like an excuse for the genes to replicate themselves? Does that picture change a lot with this kind of thinking, or is the fundamental philosophy the same and this is just a new mechanism?
Now, of course it changes. I mean, it's not the genes, we are not survival machines for the genes. The genes is an input into development. It's an input into development. It's a hereditary input into development. Change variations in DNA sequence are inputs into development, into heredity and development, as are epigenetic changes, as are cultural changes. They're all inputs into development and heredity and evolution. So it's a very different kind of view.
So instead of the evolution working around the gene, it works around the evolving organism.
Yes.
The developing organism.
Yes, exactly. It's the organism in its world that is the center, not the gene. And that's a very, very different way of looking at it.
Okay, that was it for today. Thanks for sticking with us until the very end. And it was the last of these little bonus clips of unheard sections from last year. Within the next week, you can access the first new full-length episode of 2025 that will be on the origins of patriarchy. So stay tuned. Until then, take care.